Hello, OHBM. My name is Nicole Eichert, and I'm thrilled to present our paper to you. I find the hippocampus a very interesting structure to study in the context of brain evolution. It is typically considered conserved across species. And when you look at such a histological cross-section, you will also find the same canonical pattern of what we call hippocampus subfields across the species. But at the same time, we know the hippocampus is involved in many higher order cognitive functions, such as autobiographical memory. So we appear to have both conservation and innovation represented in the same structure. Resolving this tension was the aim of our project. And to do so, we first developed a comparative framework to study the hippocampus across primates. We used a new tool called Hippocampal and adapted it for the macaque brain. As you can see here, it reconstructs a hippocampal surface, for example, from an MRI scan, and then it computes the coordinates of the intrinsic axis of the hippocampus, so anterior-posterior and proximal-distal. Based on these coordinates, we can then represent the same surface as an unfolded or flattened map. So we unfolded the macaque hippocampus, and importantly, we did the exact same operation in the human brain. So how do they compare to each other? We wanted to start out by looking at a set of geometric indices in this flat map space. And what we find is that the flat mapping achieves a very good matching between species. For example, you can see here a hotspot in the gyrification map in the human that represents the point where the head of the hippocampus curls upwards. And the equivalent point in the macaque brain is represented in a very similar location. So what this means is that this surface and the flat map space is matched for geometry. It also means that every point in this map in the macaque is directly equivalent to the same point in the human. So what about histology? We accessed the histological data from the Big Mac resource and manually labeled the hippocampal subfields. And then we mapped all this information to the surface, resulting in this flat map of microstructure where subfields appear as vertical stripes. In this 3D visualization, it might be easier again to understand how this flat map relates to the underlying anatomy. And we actually performed the same mapping with some raw intensity values of the histology and also some MRI-derived microstructural markers. And they all recapitulate the same classic subfields. And now, again, importantly, the exact same procedure has previously been performed in the human based on the big brain resource. And when we look at the resulting maps of both species next to each other, we can clearly see that they have a very similar pattern. So overall, the hippocampal microstructure appears to be conserved, at least across those two species. And what this result also means is that our 2D flat map space actually represents a common space to compare the two species. And having for the first time such a matched coordinate system also allows us to do spatially resolved quantitative comparisons. For example, we found that the width of the CA2 subfield in the human is wider than in the macaque, which fits in with existing hypotheses. But these kind of quantifications will need further validations in a larger sample. For our purpose, we leveraged this new comparative space for functional connectivity analysis. Based on in vivo resting state MRI data, we employed some familiar steps of nonlinear connectivity embedding. And when we look at the resulting primary axis of variation, we find that it varies along this long hippocampal axis, and that's true for both species. So again, the overall functional differentiation along this axis appears to be conserved across species. But having such a shared coordinate system in the hippocampus allows us to do more than this. We can actually use the hippocampus as a shared feature space to implement a joint embedding combining both species together. And when we apply such a joint embedding, we find that the primary component reveals a network that reminds us very much of the default mode network. And by nature of the input data and the way it is constructed, we know the two maps in the two species are actually comparable and homologous. In the human, this network is well established, and in fact, if we do a regular cortical embedding of resting state data, we are able to recover this network very well. However, in the macaque, such a cortical embedding reveals this network to be split into two separate components. And typically these two components, or only one of them, have been reported as the default mode network in the macaque before. But what we show here is that if you anchor your analysis in the hippocampus, you can reveal the entire network being comparable. So we conclude that the hippocampus in both species is embedded in the default mode network. And finally, we wanted to ask, how do these intrinsic hippocampal axes, anterior-posterior, proximal-distal, how are they themselves reflected in brain connectivity? And we studied this in a dual regression approach, where we used those hippocampal axes as orthogonal spatial regressors. And when we look at the results, we see that both of the axes relate to the default mode network, but with slightly different contribution. In a further targeted quantification, we also found that there are some marked differences between the species. So we found that the inferior parietal lobe in the human is very strongly differentiated in both axes, but this seems not to be the case in the macaque. So that suggests that the inferior parietal lobe is a hotspot for reorganization and specially wired in the human. Now, before I sum up our findings, I want to say a few words about data and code availability. All of the code and data have been made openly available. And if you have further questions about how to best use them or how to implement the hip unfold in your own non-human primate data, please get in touch with us. And we're very happy to help you with that. And now I just want to finish up by showing you couple of take-home messages that I would like you to remember. One is that the hip unfold flat map represents a novel space for comparative research, and it's matched for geometry and histology. We found that the macrostructural organization, the principal functional axis, and the cortical networks of the hippocampus are overall conserved across primates. And what we also found is that the brain-wide embeddings of the functional connectivity data also reflected evolutionary reconfiguration of higher order networks, in particular the default mode network. And specifically, the inferior parietal lobe in the macaque mirrors the incomplete integration of the DMN in non-human primates. And with this slide, I would like to conclude my presentation and thank my co-authors and my collaborators. I'm very sorry I can't be there with you today in person to answer your questions, but I hope you enjoyed the presentation nevertheless. If you want to reach out and talk about the material, please do so. I'd be very happy to talk to you. Thank you very much.